Now let's look at some cells that help to defend us non-specifically. These cells need to be capable of identifying non-self cells. We don't want our defensive cells just to kill any cell they bump into because then they'd be killing our own cells. So these non-specific cells need to be able to recognize a pathogen as a pathogen. And they're able to do this by having certain receptors embedded in their cell membrane. These receptors are on the surface of the cell, and they can recognize certain features that are common to many different pathogens. For example, most bacteria have a cell wall with something in it called a proteoglycan, which is a protein-sugar combination. So there's a receptor that recognizes this proteoglycan, and that's how we can tell that this is a pathogen. So our cells don't contain that proteoglycan. Even though we're using a receptor here, this is not specific. The receptor doesn't recognize a specific pathogen, right? It just recognizes, okay, this is a bacteria. It can't recognize that this is E. coli bacteria or Salmonella bacteria. So this is still considered nonspecific. These receptors are called pathogen recognition receptors, which makes sense because it helps the cell to recognize the pathogen. So basically, these pathogen recognition receptors read the outer surface of other cells, checking to see if it's a self cell or a foreign cell. Healthy self cells have certain identification markers on their surface that our defensive cells recognize and leave alone. Pathogens contain, as I said before, these certain structures on their surface that are foreign, easily recognizable as foreign, and pathogens also lack self-identification markers. So those two things combined make it pretty easy for our defensive cells to recognize a pathogen as a pathogen. So basically our defensive cells bump into the pathogen and then kill them. And they usually kill them by taking the pathogen into themselves and then digesting it, basically using it as food. This process is called phagocytosis. It's basically the cell eating something. To give you a specific example of a pathogen recognition receptor, we have something called a toll receptor. And this is just one of many different types of pathogen recognition receptors. Let's take a look at this process in a more pictorial format. So on the screen here, we have a self cell, and these stars are representing self-identification markers. So all of our self cells are going to have these stars. This is a nonspecific defensive cell, an innate immune system cell. And you can tell it also has the self-identification markers on it. Additionally, it has these U-shaped things that are representing the pathogen recognition receptor. Over here, we have our pathogen. So the self cell comes along and bumps into the defensive cell and nothing happens. And that's exactly what's supposed to happen. This is a healthy self cell. So our nonspecific defensive cells should ignore it and do nothing. But this is a little different when it comes to the pathogen. When the pathogen comes along, our defensive cell recognizes these green things on the surface. The green things on the pathogen are representing surface molecules on the pathogen that are common to lots of different types of pathogens. So this is something that's easily recognizable by the pathogen recognition receptor. So once the two bump into each other, the process of recognition occurs, and then this is going to signal for the immune system cell to phagocytose the pathogen. Once the pathogen is inside of the immune system cell, it's broken down or digested, if you will. So essentially, the pathogen's been destroyed. Now let's look at some examples of the cells that help to defend us nonspecifically. These include neutrophils, dendritic cells, macrophages, eosinophils, basophils and mast cells, and natural killer cells. Neutrophils are a type of white blood cell, or leukocyte. And of course, that would suggest that neutrophils are found in the bloodstream, which is true. Neutrophils are in the bloodstream, but they'll move towards an area of the body where there's an infection. But how does the neutrophil know where the infection is occurring? Well, that's because of chemicals called cytokines. So cytokines are chemical messengers that are released by damaged cells and other defensive cells. And the cytokines act as a chemical communicating system, alerting the body to pathogens and the possibility of infection. Neutrophils migrate towards high concentrations of cytokines, and this is called chemotaxis. So chemical movement, that's what chemotaxis means. So the chemicals are causing the neutrophil to move towards where the chemicals are being released. The cytokines also serve to activate neutrophils. Neutrophils will work without the cytokines, but basically cytokines are going to cause any type of defensive cell to kind of work a little bit better. Once the neutrophil is activated, basically we have three different options. The neutrophil can go ahead and phagocytose the pathogen. So gobble up, eat it, break it down. The neutrophil can release defensive compounds into the general area where the pathogen is present, and these compounds can help to destroy the pathogen. The third way the neutrophil can help to defend us is actually really cool, and this is the fact that neutrophils can actually set pathogen traps. They release substances that are basically kind of sticky, and it prevents the pathogen from moving around in the area and kind of makes the pathogen kind of collect all in a certain area so that the pathogen can't travel or grow anymore. Macrophages, like neutrophils, are also a type of white blood cell. And like a neutrophil, the macrophage is also capable of recognizing pathogens and then phagocytosing them. But macrophages are much better at this than neutrophils. So macrophages are our main non-specific cell that we use for phagocytosis purposes. Like neutrophils, the macrophages are also activated by cytokines. So cytokines are these general chemicals that are used to kind of activate the immune system. Macrophages are actually going to be really important when we look at our specific immune response. And that's because macrophages are going to act as a messenger between our non-specific and specific defenses. In the picture, we can see a macrophage that has sent out extensions of its cytoplasm in two different directions in order to phagocytose bacteria. Dendritic cells are very similar to macrophages in terms of their function. The difference really is basically location. Macrophages are going to be found in most of our body tissues and also in the bloodstream, whereas dendritic cells are really mostly isolated in our tissues that have some sort of contact with the external environment. So this is going to be places like skin and our mucous membranes. Like neutrophils and macrophages, the dendritic cells are also activated by cytokines. Once activated, they can easily gobble up lots of bacteria. Dendritic cells are much more rare compared to macrophages, and they're very hard to isolate for research studies, so we don't actually know a lot about dendritic cells. One of my favorite non-specific defensive cells is the eosinophil. Eosinophils, like neutrophils and macrophages, are a type of leukocyte, so they are a type of white blood cell. And like those other cell types, they are also activated by cytokines. However, they don't phagocytose bacteria. Their specialty is fighting parasites. In the cytoplasm of the eosinophils, there are enzymes that can be released onto the surface of parasites that will cause them to break down. And if you follow the link in this video here, you can watch a short video of eosinophils attacking a worm. And it's really cool. They speed it up real fast, but basically you see all these eosinophils come over to a worm, they release their chemicals, and then the worm does like a death spasm. I put basophils and mast cells together because they are very similar. They do have different origins in terms of basophils are a type of white blood cell and mast cells are not. However, when they develop into their final form, they are nearly indistinguishable. Both basophils and mast cells are very important for the inflammatory response that we'll talk more about in a little bit. The way basophils and mast cells aid in the inflammatory response is by releasing two important chemicals, histamine and heparin. Histamine you've probably heard of because you've probably taken an antihistamine when you've had some sort of like allergy attack, or maybe you have seasonal allergies. Histamine is a chemical that promotes blood flow, and it does this by causing blood vessels to dilate. And when a blood vessel dilates, you get more blood coming in the area, so that means more liquid. And then as the blood vessel dilates, it becomes leakier. So that means more fluid can leak out of the blood vessel into the surrounding tissues. And this is important during infection because it helps to bring more defensive cells to the area where the infection is. Heparin is an anticoagulant. What that means is that it prevents blood from clotting. So this allows blood to flow towards the infected area, bringing defensive cells and healing chemicals and healing cells along with it. Basophils and mast cells are not our friends when it comes to allergies, though. These are the two cells that are very problematic with allergies, and we'll talk more about these later. Natural killer cells are also a really cool type of defensive cell. What makes them so cool is that their job is to hunt down their own kind, so to speak. Natural killer cells have the job of recognizing self cells that are either infected or cancerous, and then the natural killer cells will kill that particular cell. So I like to say that natural killer cells kill our self cells that have gone rogue, so to speak. Infected cells and cancerous cells often have an increased level of a certain type of self-recognition marker called major histocompatibility complex, better known by its abbreviation MHC. So this is a protein that's on the surface of all of our cells, and it's increased when a cell, like for example, is harboring a virus or a parasite, or if it's cancerous. And the natural killer cells can recognize this high concentration of MHC. When they recognize this, the natural killer cells then secrete a protein called perforins. And perforins basically poke holes in the cell membrane of the infected or cancerous cell, making it very leaky, and eventually it dies. So the cells we just talked about are very important in providing a first line of defense against pathogens while we wait for our specific immune response to kick in.